A monohybrid cross is one in which two individuals that differ in the two traits of one character are mated and produce offspring. In order to understand how a monohybrid cross works, we need to diagram it from the parental generation to the F1 and then the F2 generation. Let's say that we breed two parental individuals that are both homozygous for a particular trait. In this case, we'll call it A, where the capital A is the dominant trait or dominant allele, and lowercase a is the recessive trait or the recessive allele. In order to cross these two individuals to determine what the F1 will look like, we first must take each of the parents through meiosis. Remember that in meiosis, the chromosomes separate from each other to produce haploid gametes. Following Mendel's law of segregation, we can use the alleles in each of the individuals to represent gametes. So if meiosis were to occur to each one of these individuals, the alleles would segregate from each other, producing individual gametes, each of which only contains one copy of the allele. Now that we have the gametes identified for this cross from each of these parents, we can set them up in a Punnett square, which is a tool that can be used to determine what the offspring of a cross between two parents will be. A Punnett square in this case is a 2x2 box in which the alleles from each parent are written down on the top and the left side of the box. We will do that as so. On the top we have the two alleles from the dominant individual, and on the left we have the two alleles from the recessive individual. Remember that these letters represent gametes, and when gametes fertilize, they will produce the offspring. So what we do with the Punnett square is we fill in the boxes, placing both alleles together. Notice that the capital letter or the dominant allele is always written first. That is a basic rule of genetics. All of the individuals that we see in this box, you'll notice, are heterozygous. These individuals represent the F1 generation. In the cross that we did, where both individual parents were homozygous for both for either the dominant or the recessive allele, a cross between those two will always produce a heterozygous F1. Now let's cross the F1s together. We take one F1 individual, write its genotype down next to the others with an X in the middle, which you saw earlier. That represents the crossing. Now, we have to figure out the alleles for these two individuals that are going to be represented in the gametes. So again, going through meiosis, these will segregate independently from one another. And now you'll notice that each of these individuals produced two different gametes, one with a dominant allele, capital A, one with the recessive allele, lowercase a. Our Punnett square now will look a little bit different, but we'll use the same basic format. It will be a 4x4 or 2x2 square with each individual's gametes or alleles on either side. When the gametes fertilize, we fill in the square, and what we have here are the offspring that we will now call the F2 generation. Recall that in the F1 generation, all of the individuals were heterozygous, therefore exhibiting the dominant phenotype. If we go down now to the F2 generation, notice that 3 out of the 4 show the dominant phenotype, and 1 out of the 4 shows the recessive phenotype. This is typical of a cross like the one we did, where the two parents are both homozygous for each trait, one homozygous dominant and one homozygous recessive. So what we see then is we have a 3-to-1 ratio of the dominant to the recessive phenotype.